All right, so now we're going to kind of zero in on the cell and talk a little bit about cellular injury. And sometimes these mechanisms are, important to understand and distinguish. And so we'll kind of walk through this process of what happens to a healthy cell, the early findings that we see with cellular injury, and then some of the late findings. So just to start this conversation off, I just kind of want to walk you through some of the things that are happening in the cell to kind of make sense of what happens during cellular injury. So there's a couple different things happening here. So first, the first thing you have to keep in mind is that oxygen, I'm just going to write O2 here, this oxygen is required to be taken up by the electron transport chain as the terminal electron acceptor. So this oxygen can then assist in the formation of ATP, which is exactly what we see here. So oxygen, they're going in the mitochondria, right, supporting the electron transport chain, and then we have the formation of ATP. Now this ATP is going to be critically important for things like a lot of the pumps here on the surface. So the classic one is your sodium potassium pump. Okay, and this is an ATPase. So it uses ATP, breaks down ATP, and uses that energy to pump these ions against their gradient, right? And so you have sodium that is getting pumped out of the cell, and then you have potassium that's being pumped in the cell. Again, both of these are being pumped against their gradients. Now this sodium, once this sodium is in the extracellular space, this sodium can then be exchanged again for calcium. Okay, so we're kind of, in a way, indirectly pumping calcium out of the cell by using the sodium potassium ATPase. That's one way you could look at it potentially. And so sodium, again, is getting exchanged here for calcium to get pumped out of the cell. Okay, so we have a sodium calcium exchanger. And then on the bottom here, we also have a calcium ATPase. So this is another way that we can pump calcium out of the cell by using ATP. Okay, so we also have a calcium ATPase. So you can see there's multiple ways to get this calcium out of the cell. And so, you might be saying, well, that seems pretty important, right? So the idea here is this calcium, we don't want it to sit in the cell, because if this calcium sits in the cell, what will happen is the calcium can set off a lot of different cascades that eventually lead to destruction of the cell, okay, by activating lysosomal enzymes, endonucleases, and those are all the things that we're going to talk about here. And you can see when this process goes, wrong, essentially, when there's something that's not working in this process, particularly when there's hypoxia, right? If there's no oxygen, we can't make ATP, then all of these downstream effects will lead to an increased level of calcium. And the reason being because I can't get any of this calcium out. Okay, so we're going to walk through that. So again, we're talking about the early reversible stage of injury. So let's just say that we had a patient that, has some ischemia to some tissue, right? So if this patient has ischemia, whether it's a myocardial infarction, a stroke, whatever it is, there's some ischemia going to a portion of the tissue. And so the oxygen that's getting to that tissue is going to be lower, right? And so we would say that this patient is undergoing some form of hypoxia, right? And so when this happens, we already know the first step, we said the oxygen is primarily going to be used by the electron transport chain to generate ATP. So we're going to get a lower level of ATP, likely because we don't have our terminal electron acceptor from the electron transport chain, right? So that ATP that drops is going to have a few different effects, right? So for one, we're going to get inhibition at the sodium potassium ATPase, right? And then we're also going to get inhibition at the calcium ATPase. And then indirectly what's happening is my intracellular sodium, right? My intracellular sodium is going to go up because I can't pump it out because I don't have ATP, right? So my intracellular sodium levels go up, and my intracellular calcium levels will also tend to go up for the similar reasons, right? My intracellular calcium will go up because I can't exchange it for sodium because I'm not pumping the sodium out to begin with, and I don't have ATP to pump calcium out through the calcium ATPase. So for those reasons, the intracellular calcium will go up. And so this is kind of what we see here in the text here. So again, hypoxia in each of these situations is going to lead to a decrease in ATP because of the lack of oxygen, right? And so that's going to eventually lead to decreased function at the sodium potassium ATPase, increase in intracellular sodium, increase in intracellular calcium. Same concept here, right? I don't have ATP for the calcium ATPase that's down here, and for that reason, the intracellular calcium levels are going to go up. And finally, the last point here that we didn't really talk about yet is this decrease in oxygen is going to force us to go down a different biochemical pathway. And so that's going to lead to an increase in lactic acid as we kind of proceed down glycolysis if we can't go down the TCA and electron transport chain because of the low oxygen levels. Okay, so we get an increase in lactic acid for that reason, and this lactic acid can also contribute to protein denaturation. Okay, so you have multiple kind of factors working here in the setting of ischemia. And so if we take a look at our image here, we can see these red X's indicating, look, if we don't have oxygen, right, I can't generate ATP. I can't get through this first step. That's going to inhibit, like we said, the sodium potassium pump. So we see all this sodium sitting intracellularly. Can't get this calcium out. It's sitting intracellularly, right, because I don't have ATP and I don't don't have sodium here in the extracellular space, or enough sodium potentially to swap sodium for calcium. Okay, so what you start to get, you'll start to see this kind of irregular shape to the cell. Okay, and we call this plasma membrane blebbing. Essentially, what's happening is a lot of the phospholipids that are sitting in the membrane here are going to start to get destroyed. And this is largely due to the disruption of this gradient that we had, right? This electrochemical gradient is getting disrupted. We're getting damage to the phospholipids. We also have high levels of calcium intracellularly. That's leading to the activation of a lot of these different endonucleases and lysozymes, right? And so these cells will start to get some destruction, and eventually we'll start to leak our cytosolic enzymes out into the extracellular space. So theoretically, right, let's just say that this was a cell of the heart, right? So let's just say this is a myocardial cell. Okay, so this is a myocardial cell, and let's just say that this is the cell that has this damage. Maybe it's due to ischemia, right? We have a patient that maybe has angina or has some form of acute coronary syndrome. Okay, so what we know is that these cells tend to have troponin in them, right? And so this troponin, right, as this damage happens in the phospholipid membrane and as this cell starts to bleb and swell, we'll eventually see this troponin, right? As these phospholipids kind of get damaged and they swell and they bleb, this troponin will start to leak, right? So this troponin will start to leak out into the blood. So now that this troponin is out of the cell, right, if I do a blood draw on this person, right, now that this troponin is in the blood, out of the cell, if I do a blood draw on this person, right, here's my needle, I draw some blood, I might see an elevated level of troponin in the blood. And that elevated troponin is telling me that there's potentially some myocardial cell damage because I'm leaking the enzymes of that heart tissue. Conversely, right, if this was muscle tissue, maybe it wouldn't be troponin. Maybe I'd see an elevation in my CK, my creatine kinase, right? The CK level might be up if it was muscle tissue. And so we have different markers that would tell us which cells potentially are damaged. These markers are what we would expect to normally be inside of those cells. It's just that we have some damage in those cells, and those cells are now leaking some of their enzymes and proteins. All right, some other findings here, largely due to the lysosomal activation as well as potentially lactic acid, is this ribosomal detachment. And so here you can see the ribosomes are nice and neat on the rough endoplasmic reticulum, and here you can see the ribosomes are kind of now detached from the endoplasmic reticulum. And this is another very kind of classic sign that you'll sometimes see mentioned for early stage injury. We can also see mitochondrial edema. Now, the point that I want to make here, though, is that generally, and this is going to vary a little bit in terms of cell types, but just as a general rule, early stage injuries tend to be reversible. So the point being, when I say reversible, if suddenly now I start to give this patient, and I realize this patient has ischemia to their heart, and we do a coronary catheterization and open that area up, right? Or if we put a stent in there, right? And all of a sudden now this area of the heart can get the oxygen that it was deprived of. Well, in these situations, now we can start to turn around this ATP production and try to reestablish these electrochemical gradients. Even though I may have leaked some of that troponin, it doesn't mean that those cells are going to all completely get destroyed, right? But if we wait, right, and we don't do anything and we don't get this oxygen to that tissue by doing a coronary catheterization or procedure, then what can happen is we can end up with an irreversible injury. And different tissues will have different time frames that they can, go through ischemia before they get to this stage. So this is going to be variable. But the point is, once you get to this late irreversible stage, what eventually happens is you have so much intracellular calcium that builds up, for the reasons we talked about, that you start to activate a lot of those enzymes from the lysosomes. Again, these destructive cellular enzymes are largely going to be released from the lysosomes. So this process of the lysosomes destroying the cell that they're within because of this very high calcium level is called autolysis. And as we said, as this is happening, there's also destruction of the plasma membrane, and so we're going to leak more of those cytosolic enzymes. So that troponin level might get a lot higher, right, as this patient starts to infarct tissue and gets to this irreversible stage of injury. Okay, now the thing that happens here, and we're going to have a whole video on this, but eventually what will happen is when you start to get into this late stage, these cells are essentially programmed to say, look, there's been so much damage around here that we can't clean this mess up. We're going, one way down this road. We're at a point where we're at irreversible injury. And so what the mitochondria will do is they'll tend to increase the permeability, kind of like we see in the cell wall. As we transition into the late stage, this mitochondria will start to leak out cytochrome C. I'm going to put site C here, but that's for cytochrome C. And so similar to what we see with cytosolic enzymes leaking into the extracellular space, this cytochrome C will leak from the mitochondrial space into the cytosol. And what this cytochrome C will do is it will eventually trigger apoptosis of the cell. Now, apoptosis is basically, as we see here, programmed cell death. So this is kind of like a trigger for the cell to say, hey, look, something is wrong. We need to kind of shut things down because at this point, there's irreversible injury. You don't want cells that have injury, like the cell we see here, or a late stage injury especially, you don't want those cells to go on to replicate, right? You want to shut down the cell cycle, particularly if, there was injury to a cell and the injury was due to a mutation, right? If there was a mutation in a cell that completely threw off all of these transporters in the mitochondria, you don't want that cell replicating because that cell will carry that mutation to the daughter cells, and that's how you end up with cancer, essentially, right? Or neoplasia, all right? And so that's why we have this signal, this apoptosis. So when we get to this point, the mitochondria can release cytochrome C, and this cytochrome C will eventually lead to apoptosis of this cell. Now, what's important to understand with apoptosis is a couple things. One, when we have this signal from cytochrome C to trigger, and we'll talk about this, to trigger the caspases, these caspases that lead to programmed cell death, they don't cause inflammation around the tissue. We could potentially have other things that cause inflammation, but again, this process of apoptosis in and of itself should not cause inflammation. And generally with apoptosis, what we see is this cell membrane, it actually stays intact. So in other words, if you had a genetic defect that caused there to be an issue with your transporters, usually the cell membrane will not get completely destroyed before apoptosis occurs. Now with necrosis, on the other hand, it's a little bit different in the sense that this is more of an unprogrammed cell death. In other words, the cell is being destroyed not because it has a signal to undergo destruction. The cell is being destroyed because it physically cannot sustain the requirements needed to exist. Right? And so this could be because we have complete disruption of the cell membrane, right? If I had complete disruption of the cell membrane, I start to leak contents out here, I start to have contents come in, this cell is going to swell very quickly because I can't keep an electrochemical gradient. And so we might get some significant cellular edema, potentially some inflammation around this cell that's leading to, the destruction of the cell. Okay, so big points for necrosis. We do see disruption of the cell membrane, and we do also see an inflammatory reaction classically. And there's all different types of necrosis. We have a whole video on apoptosis and necrosis, so we'll go into more detail on those two there. But just remember, apoptosis is physiologic, right? Necrosis is pathologic in general. So for example, if you're ever in a situation where you have to distinguish these, if you see things like granulation tissue mentioned, right? So granulation tissue, or if you see, a bunch of neutrophils around some cells and you're asked, is this necrosis or apoptosis? Well, if neutrophils would signify that there's an inflammatory reaction, right? Granulation tissue might be representative of chronic inflammation, or in general, if you just see really significant cellular edema. Okay, this could be because the cell membrane is destroyed. So these kind of findings in general are going to be more consistent with necrosis. Apoptosis, usually there's going to be some programming in the cell that basically shuts things down before we get to that point. The last thing here are these stages of, nuclear condensation, fragmentation, and dissolution that happen towards the end of this irreversible stage. So these are findings you'll see in the nucleus as we kind of get to this late stage where we start to get disruption. And, this is a great image. This image, if you saw this image, right, were you thinking more necrosis or you're thinking more apoptosis? Well, again, the fact that you have complete disruption here of the cell membrane, this would be significant for necrosis, most likely, as opposed to apoptosis. But again, going back to these findings here at the nucleus, so the first thing we'll generally see in this late irreversible stage is pycnosis. And this is essentially nuclear condensation. So nuclear condensation, and what that means is condensation, you're thinking about the nucleus coming together and essentially shrinking. And so that's kind of what you see here. You can see the nucleus getting smaller. It's essentially shrinking, right? So we're shrinking the size of the nucleus, and this is an example of pycnosis, nuclear pycnosis. And you might be saying, do we see this finding with apoptosis or necrosis? You can see nuclear pycnosis with either one, right? Either one. And the bottom line is, once you see this finding, this is the important part. Once you see this, this tells you that you're going down an irreversible path. Okay, so when you see nuclear condensation or pycnosis, that's telling you this is irreversible late stage injury. So that's also important to remember. This one is pycnosis. Now, the next step that you might see is karyorrhexis. Okay, so karyorrhexis, I just think of the word, rhexis, just like ripping apart the nucleus. That's this one here, karyorrhexis. Okay, so we're essentially kind of fragmenting the nucleus. There's the more formal definition, nuclear fragmentation, right? Or just kind of ripping the nucleus, right? That's karyorrhexis. And the final one on here is karyolysis, nuclear dissolution. So once we've kind of broken this nucleus up into pieces, we kind of just see it start to dissolve, and we have some disintegration of the nucleus. Okay, so it just starts to kind of dissolve. Okay, and this last step is called karyolysis. So again, karyolysis representing the dissolution. So pyknosis, we start to condensate, then we see it kind of broken up into pieces, that's karyorexis, and then we see the dissolution. Again, all of these happen. When these happen, it's basically telling us that, again, it's irreversible. When we see these findings, we're at a late stage or irreversible stage. And again, you can see this in apoptosis or necrosis.